Hello, my name is Joshua Mugabi. I welcome you to Crotalaria Flower. A Crotalaria flower, this is how it looks like. It has standard petal, it has a bud, and then the side flowers as we are going to see it. We have the standard petal, this one up, and then it has got a petal which looks like a bud. This one here, as you look at it, and then it has the wings, petals which look like wings. There are two, one this side, another one the other side. Then it has got 5 sepals which are joined together. So when you look at it from the front, this is what you are going to see. You will see a standard petal, 2 of them joined together, and then it has got the wings, petals which we call the wings, and on the bottom it has what we call the keel petal, which looks like a bud. So here you have the keel petal that passes through the wings. Here is what we call the bud shaped, and then the wings, they are the one on the side, one on this side and another on the other side, as you can see here, the bud here and the bud this side. Then we have the one which is outside, which is fused, which we call the standard petal. And then we have the sepals, which are 5 sepals. So when you look at it, you will see the standard, the wing, and then the keel, and then the sepal. Using a razor blade, cut the specimen longitudinally. So we shall get the specimen and cut it with a laser blade through longitudinally. Draw and label 1/2 of the specimen. So when we cut it through, it means we shall cut through the standard, then we shall cut through the bud or the keel, and then on one side we'll go with 1/2 of the standard, 1/2 of the wing, and 1/2 of the one of the wings and 1/2 of the keel. Then we shall cut through the ovaries and then the stamens and the anthers and style. So using the razor blade, we have cut through the specimen longitudinally, and now we are supposed to draw and label 1/2 of it as we are seeing it here. Now when you have cut it through, this is what you are going to see in the 1/2. We have the banner, which we call a standard flower. We have the wing. This is the wing or petal. And then we have the keel. The keel is this one, which is bud shaped inside. And then we have the stamen, made up of anthers, group of anthers, and fuse this stamen. This one is a free stamen, one is free, and then the other ones are fused together. And in the middle you will see the pistil. And then we have the calyx, which is made up of sepals, and the pedicel. So that is 1/2 of the Crotalaria flower cut through longitudinally. So here it will show you different parts. This one we call it the standard petal, and we have this and this one, they are wing petals. Then you have this one in the middle. This one is what we call the keel, which is bud shaped. When you remove it, this is how it appears. It appears like a bud, and It has two fused petals. There are two fused petals, but inside it, it is enclosing the ovary and the stamen. Then we have the pedicel. The pedicel is this one. If you remove the pedicel with the ovary attached, you will see that there are 9 stamen, and then we have the ovary down. We shall have the ovary down, and then the stamen, and then we shall have the style and the stigma on the tip. That is how our Crotalaria flower looks like. The ovary becomes a pod containing 2 to 3 seeds called peanuts. So this is how it appears, our drawing. We have the standard, we have the sepals, there are 5 of them. Standard, remember it is joined, there are 2 joined together, fused together. On the sides we have the wing, and then the boat-shaped one is what we call the keel. Then we have in the middle the stigma and the style, and then we have the ovary, and then we have the filaments with the anthers. That's how our drawing would look like. So here you can see it very well. You have the standard, which is fused. You have the wing on one side, and then on the other side you have another wing, and then you have the boat petal, which is passing through here. That is the boat petal, and you have the calyx, or a group of sepals fused together, 5 of them. Describe the sepals of the specimen. Describe the sepals of the specimen. You see that there are 5. We have sepals. There is 1, 2, 3. 3 are the ones that are seen. There are 2 the other sides. There are 5 in numbers, and when you look at them, you will see that they are fused together. So the sepal is fused with this one, and the other one is fused with the other one. They are fused together, and they are green in color. If you look at them, you will see that they are green in color, and they are tapering at the apex. When they are coming to the apex, they taper. You can see that they are tapering at the apex. And they are hairy. They have hair around them. They are veined. They have veins. If you look at them, they will be having veins. You will see veins there. Describe the petals. Describe the petals. There are 5 petals. We have the standard. We have 2 wings, and we have the keel, 5 in number. 3 are free, and 2 fused. We have 3 here, this one and this one. 1, 2, 3. They are free, and then we have the standard has got 2 which are fused together. Standard curves backward and exposes the honey guide. When you look at the standard, this is the standard. It curves backwards and it exposes the honey guides. Honey guides, they are these lines that you are seeing in the standard. Those are the honey guides on the standard. And the wings are hooked to the keel to expose the stigma. So we have the wing, this is the wing, the other one is the wing. So when they are hooked, they cover the keel, but they expose the stigma. What you're seeing outside here, which is coming out, is the stigma. So when you look at this one, you will see that this one here is the wing, and the other one is the wing on the other side. So they are hooked to the keel. This one is the keel inside, but they are exposing outside, they are exposing the stigma. The keel is boat-shaped to enclose the pistil and the stamen. So the keel, this one, for it is like a boat, and inside it, it is covering the pistil and also the stamen. Narrow at the base. All of them, they come and they narrow at the base. They are smooth and veined. The petals are smooth and they have veins. Describe the stamen of the specimen. Describe the stamen of the specimen. Now here we have our flower, different stages. Here it is covered in the calyx, sepals. The sepals are opening and the flowers are coming out, and then here we can see the flowers are already out. We can already see the standard bent upwards or forward, and then it is leaving the wing, the other side, covering hooked over the keel. Now when we remove the sepals and we remove also the petals, then we can get the male part, the flower parts inside the stamen and the style. They are 10 in number. If you look at them here, they are 10 in number. The anthers and the filament, the stamen. The one which you are seeing, which is long, that one is the pistil, the female part of the flower. But these small ones that you are seeing, those are the stamen. They are 10 in number. 9 filaments join to form a staminal tube. 9 of them, they become fused. 9 of them, they are fused to form a staminal tube, and 1 of them is free. They're shorter than the stigma. If you look at them, they are very short compared to the stigma. This is the pistil, and pistil contains the style and then the stigma. The stigma is on top. And if you look at the stigma is above the filament or the stamen. It is above the filament, meaning that the stamen are shorter than the stigma. Describe the pistil. Describe the pistil. It has 1 couple. When you look at it, this is the pistil, the female part of the flower. It has 1 couple, that is the ovary is 1, and then it comes and forms the style and then the stigma up. And it has marginal placentation. You can see that the placentation is on one side, margin, and the ovules are attached on the margin. The placentation is marginal, and the style bends upward. If you look from the ovary, you come to the style, and the style is going to bend upward, moving upward. That is our style, meaning that it is curved and it is superior. When we say that the pistil is superior, it means that the ovary is above the petals. When you look at its location, the petals are below the ovary, so the ovary becomes superior. So this is a simple exercise. You are provided with the specimen 2, which is a hibiscus flower, and specimen R, which is a Crotalaria. Look at them and try to look for the differences between the 2 specimens. So state 4 structure differences between the 2. So when you look at specimen 2, which is hibiscus, it has several lines of symmetry. Lines of symmetry means that you can cut it through here, cut it through here, cut it through here, and it will be the same. Whereas if you look at the Crotalaria, it has only 1 line of symmetry. You only cut it through longitudinally like this to get sides which are equal. When you cut it through the other angles, they are not going to have equal parts. Then the stigma is exposed. When you look at the hibiscus, the stigma is exposed up, and here the stigma is enclosed. You cannot see the stigma, it is enclosed. Then when it comes to the pedicel, this one has got a longer pedicel and this one has got a shorter pedicel. When it comes to the petals, these has fused petal at the base. They are fused at the base, but when you look at this one, the petals are free. The petals are free here. They are not fused. This one has got an epicalyx, that is hibiscus. It has got an epicalyx, other sepals. Before the sepal, we have the epicalyx, and the Crotalaria has no epicalyx. It has exposed stamen. The stamens are exposed. When you look at them here, they are exposed, and these ones, the stamen are enclosed. Here the stamens are enclosed in the keel. This one has got a longer pedicel. It has a longer pedicel, and this one has got a shorter pedicel. Thank you very much for participating in the Crotalaria Flower. Please do like, share, and subscribe.